So, welcome all in today's lecture. In my previous lecture, I had discussed the classification of essential mineral elements. Now, one of the essential element is nitrogen, and it is required by the plants in large amount and approximately constituting 1 to 5% of the total plant dry mass. And nitrogen basically plays a very crucial role and in a different fundamental components of protein, nucleic acids, chlorophyll, coenzymes, phytohormone and secondary metabolites essential for plant functions. But you see, the availability of nitrogen to the plant root is therefore playing a very detrimental role for the proper growth and development of plant. You see that atmospheric nitrogen exists abundantly, but it is only accessible to plants that are capable of forming symbiotic relationship with nitrogen fixing soil bacteria. Therefore, most plant they rely on alternative nitrogen compounds for their growth and development. The primary source of nitrogen absorbed by the roots of the plant in higher plant typically include nitrate and ammonia. In these two forms, generally nitrogen is taken up by the roots of higher plants. So, what happens in agriculture, that nitrogen is generally supplied to the plants to different fertilizers containing nitrogenous compounds such as urea, ammonium nitrate, ammonium sulfate, or various forms of organic matter like compost or manure. So, in these forms this nitrogen could be supplied to the plants. But you see what happens? What happens if excessive use of nitrogen fertilizer is generally practiced? This could lead to the environmental issues such as water pollution through nitrate leaching or emissions of nitrogen oxide, which is a potent greenhouse gas. So, balancing nitrogen application in agriculture is very crucial for sustainable farming practices, ensuring the optimal growth while minimizing negative environmental impacts. So, techniques such as precision agriculture, nitrogen fixing crops like legumes, crop rotation and proper soil management are generally adopted to improve the nitrogen use efficiency and reduce environmental harm. So, in today's coming lecture, I am going to cover these aspects of nitrogen, like what is the essential role of nitrogen and uptake of nitrogen, then nitrogen assimilation, then how ammonia is assimilated. Now, specific deficiency symptoms induced by nitrogen. Now, on the extreme side, what are the toxicity symptoms of nitrogen? So, question arises that how nitrogen is taken up by the plants. So, just I mentioned earlier that there are two forms of nitrogen, that is nitrate and ammonium. These are the major sources of inorganic nitrogen taken up by the roots of higher plants. So, nitrate is generally present in higher concentration, about 1 to 5 millimole, than ammonium, which is present in about 20 to 200 micromole in soil solution of different agricultural soils. So, this nitrate is also supposed to be more mobile in soil than the ammonium, and therefore its availability is more to plants than the ammonium ion. So, most plants get their nitrogen from the soil in the form of nitrate. So, inside the plant, this nitrate changes into nitrite with the help of special enzymes called nitrate reductase. Then This takes this nitrite is further transported to specific parts of the plant cell called plastids, where it is transformed into another form called ammonium with the help of enzyme nitrite reductase. So you see that localization of these two enzymes are different. One, like nitrate reductase is localized in cytosol, whereas nitrite reductase is localized in plastids. So depending on the properties of soil, including texture, pH, moisture content, and microbial activity, the availability of nitrogen sources in the soil changes significantly across time and distance. Because of this, plants generally have very well developed mechanisms that regulate how efficiently they must absorb nitrogen in response to changes in the form and availability of external nitrogen, as well as changes in uh, nitrogen demand of the plant over the course of their life cycle. So this generally includes having several nitrogen transport system which mediate uptake at different external concentrations, as well as being able to change root system architecture to allow better exploitation of a particular soil volume or further exploration of a broader volume. You see that in generally in higher plants, there are two types of transporter involved in root nitrate uptake. These transporters generally belong to NRT1 or NR2 families, just you see in the figure, which is also known as nitrate proton symporters. So nitrate transporters of the NRT1 and NRT2 families generally transport nitrate across the plasma membrane in support with protons, which in turn requires the expenditure of ATP by proton ATPase for extrusion of proton to maintain the proton gradient over the plasma membrane. So this is the significance of these two kinds of transporter, that NRT1 and NRT2. Now, how nitrogen assimilation takes place in general. So just I mentioned that nitrate is generally readily mobile in the xylem and can also be stored in vacuoles of the root, shoots, and different storage organs. So for it's very mandatory that nitrogen to be incorporated into different organic acids, nitrate must be reduced to ammonium ion. So assimilation of mineral nitrogen into organic molecule is very complex process, is a complex process, not very, you can say. So nitrate is the first reduced to ammonium ion before it can be assimilated by plants. So the reduction of nitrate to ammonia by higher plant is accomplished generally in two mode. In the first step, nitrate is reduced to nitrite by the enzyme I must mention nitrate reductase, and localization of this reaction takes place in cytosol of the cell. In the second step, the nitrite generated by nitrate reductase is transported to chloroplast and is reduced to ammonium by nitrite reductase. So localization of this enzyme is chloroplast. Now, how nitrate is assimilated, we will discuss in detail. Just I mentioned that the cytosolic enzyme nitrate reductase is generally made up of two subunits that are identical and both have three cofactors that are covalently attached to different domains of the enzyme. The three cofactors that take part in the electron transfer from NADH or NADPH to nitrate are flavin adenine dinucleotide, which is a uh, heme containing, which is a heme generally bound to a domain which resembles to family of cytochromes, Another one is a molybdopterin, which is a molybdenum containing cofactor. So the nitrite generated by nitrate reductase is then transported to chloroplast for reduction to ammonium ion by the enzyme nitrite reductase, and generally this reaction is localized in the chloroplast of the leaves as well as proplastids of the root and other non-green tissues. So you see in the slide that the reduced ferrodoxin, which is produced in photosystem 1 during photosynthetic electron transport in the light, serves as the electron donor in green leaves. So a ferrodoxin binding domain, which is an iron sulfur cluster and a cyroheme cofactor bound to the nitrite reductase enzyme, transfer electrons from the reduced ferrodoxin to nitrite. In the roots of the plant, reduced ferrodoxin is generated via NADPH in the pentose phosphate pathway coupled with ferrodoxin NADP reductase. So this is the detailed view of sequential assimilation of nitrate inside the cell, and the two specific enzymes involved that I have already discussed, and it's very crucial for assimilation of nitrate. Now, how ammonia is assimilated? Generally, plant cells avoid ammonium toxicity by rapidly converting ammonium generated from the nitrite assimilation into amino acids. So, the conversion of ammonia into amino acids basically involves sequential action of two enzymes, namely glutamine synthetase, abbreviated as GS, and glutamate synthase, abbreviated as GOGAT. And these two enzymes are present in roots, in chloroplast, and in nitrogen fixing microorganisms. Generally, glutamine synthetase, that is GS, combines ammonium ion with glutamate to form glutamine. You see in the slide. This reaction basically requires ATP and divalent cation as a cofactor. So in chloroplasts, light stimulated nitrate reduction and improved ammonium absorption in chloroplast are generally coordinated by import of 2-oxoglutarate from the stroma and export of glutamate from the stroma of the chloroplast into cytoplasm, thus prevent high concentration of ammonium. Then what happens next? Glutamine is then converted back to glutamate by transfer of amide group, that is NS2, to oxoglutarate. You see in the reaction, which is a product of tricarboxylic acid cycle or TCA cycle. It is basically catalyzed by glutamate synthase that I just mentioned. It is abbreviated as GOGAT, also known as glutamine 2-oxyglutarate amino transferase. It is alternatively, this GOGAT is also known as glutamine 2-oxyglutarate amino transferase. So the conversion of glutamine to glutamate generally takes place in plastids which have two isoforms of GOGAT. One form generally accept electrons from reduced ferrodoxin. The other form of NADPH accept electrons from the respiration. So the ferrodoxin linked GOGAT isoform generally dominates in leaves, mainly in chloroplast of phloem companion cells, in leaf veins, whereas NADPH isoform is prevalent in roots. So in these aspects, these two isoforms of GOGAT enzyme differ. Now nitrogen can be transported in plants either as glutamate or asparagine. However, you see that there are certain legumes utilize urites and for translocation of nitrogen. The crucial factor determine the nitrogen translocation is the nitrogen to carbon ratio within the molecules. So, urites they are characterized by highest nitrogen to carbon ratio and facilitate efficient nitrogen transport. Additionally, some prokaryotes known as diazotrophs have the ability to fix molecular nitrogen from the air. This is very important. So, these diazotrophs can exist either as free living organisms or form symbiotic association within plants. So, the two most important significant nitrogen fixing symbiotic association involves rhizobium bacteria with legumes and Frankia, this is a bacteria with actinorhizal plants, actinorhizal plants. Now, once they are taken up by the plants, now what are the important functions played by nitrogen in the plants once they reach the destination? So, as I mentioned that nitrogen is a key structural part of the many essential biomolecules in living organisms. So, nitrogen is found basically in amino acids, which are the building block of the proteins, as well as in nitrogenous bases, which are very crucial for forming DNA and RNA. In addition to this, nitrogen is also present in cofactors which help the activity of various enzymes, and they are also present in this. Nitrogen is also present in alkaloids which are natural compounds with diverse effects in plants and animals. And in addition to this, it is nitrogen is also component of coenzymes which assist enzymes in their chemical reactions, and they are part of chlorophyll, that is a green pigment very crucial for photosynthesis. Nitrogen is also involved in synthesis of important hormones like indolactic acid. This is a very important role you must remember, and this IA plays a role in plant growth and development. So, what happens when inorganic nitrogen is absorbed by the plants? These are assimilated into compounds like glutamate and glutamine and becomes readily available for the synthesis of other important nitrogen containing compounds. These includes amides, amino acids, urites, amine, peptides, protein, nucleic acids like DNA and RNA, and various other nitrogen containing compounds. Nitrogen also basically helps plant in different ways as a whole plant level. It boosts the production of seeds and fruits, making them more abundant. It also enhances the growth of leaves and forage crops, making them healthier and more productive. Now, question arises how the deficiency symptom could be identified and where it is first recognized in plant. So, as I mentioned earlier that nitrogen being mobile element, therefore, what happens that older leaves first exhibit chlorosis and necrosis earlier than the younger leaves during the deficiency of nitrogen. So, you could identify deficiency of nitrogen once chlorosis develops in the older parts. But what happens during severe deficiency of nitrogen? Leaves become completely yellow and they fall off. So, generally a nitrogen deficient plant have a light green upper leaves and yellow lower leaves at the initial stage. And generally plant experiencing nitrogen deficiency exhibit stunted growth and slow growth because the lack of nitrogen generally inhibits cell division, and lateral buds become inactive. And plants facing a gradual nitrogen deficiency have noticeably thin and sometimes woody stem, and this woodiness could be due to the buildup of excess carbohydrate that cannot be used in synthesis of amino acids or other nitrogen containing compounds. So what happens? Carbohydrates that are not used in the nitrogen metabolism may also be used in the synthesis of anthocyanin, hence leading to accumulation of anthocyanin. This condition is generally reflected as a purple coloration in leaves, petioles and stems of nitrogen deficient plants of some species like tomato will get purple coloration, and various varieties of maize you will see the purple coloration due to the accumulation of anthocyanin. So, uh, in the scarcity of, or you can say deficiency of nitrogen, specific genes related to photosynthesis, chlorophyll synthesis and plastid protein production, they are generally replaced, and that lead to decreased plant functions. Whereas on the other hand, gene involved in secondary metabolism and protein degradation, they are activated. So, how these deficiency symptoms will appear in different plants, you see in this figure that uh, on this side you see this is the leaf having sufficient amount of nitrogen. On the right side, this leaf is deficient in nitrogen, that is chlorosis, and here you see that nitrogen deficient potato plant, and this plant is having sufficient amount of nitrogen. So, you see that in this potato plant the complete plant is under chlorosis. So, at this juncture you cannot say by visual observation that this deficiency is due to the nitrogen, because sulfur also leads to chlorosis. But at the initial stage, if you see that the nitrogen deficiency will appear only in the older parts. So, at this juncture you could say that if whole plant leaves in the lower side are under chlorosis, you could say that this deficiency is due to the nitrogen. But as deficiency continues for longer duration, whole potato plant is, you see, under the state of chlorosis. These are the two tomato leaves, one is having deficiency of nitrogen and on the other side it is also having sufficient amount of nitrogen. So, you see the difference between the two tomato leaves. Now, deficiency symptoms is also fatal for the plant, but if what happens if plants receive excessive amount of nitrogen. So, excessive amount of nitrogen could lead to toxicity symptoms, and therefore, what happens, excessive nitrogen generally lead to detrimental effects such as reduction in fruit size in crops, and affects the formation of tuber in potatoes, uh, and it could also lead to ammonium toxicity, including leaf chlorosis, stunted growth and finally necrosis of the leaves, which can result in the death of the plants. Therefore, nitrogen is basically required for the plant growth and development, and it is very important to ensure that plant receive the right amount of nitrogen to avoid these harmful effects associated with nitrogen toxicity. So, both toxicity as well as deficiency of nitrogen are fatal for the plants.